Good morning or good afternoon, everyone. I'm Julia Caro, Senior Editor here at GenomeWeb, and I'll be your moderator today. The title of today's webinar is Using NGS-based miRNA Profiling to Develop Signatures for Potential Clinical Applications. The sponsor of this webinar is HTG Molecular Diagnostics. Our panelists today are Dr. Dawn Baldwin, Co-founder and Chief Scientific Officer of MSKDX, Dr. Scott Tebbett, Associate Professor at the University of British Columbia and Chief Scientific Officer of the Proof Center of Excellence, and John Lucky, Senior Director of Marketing for Assays and Reagents at HTG Molecular Diagnostics. You may type in a question at any time during the webinar. You can do this through the control panel, which usually appears on the right side of your screen. Click on the Q&A box in the upper right side of the control panel. When you click on Send To, please select All Panelists. We will ask the panelists questions after the presentations have concluded. Dr. Baldwin, please go ahead with your talk. We just lost our audio. Just give us a couple of minutes to fix that. Thank you.
Now I can hear you. Hello.
We can hear you. Please go ahead with your talk. Thank you.
Okay. Sorry about that, everyone. My audio has been cutting out. If you lose me, I would just ask GenomeWeb to indicate that my voice isn't coming through on the chat panel. All right, so hello everyone and welcome to our webinar. I appreciate the chance to talk to you today about two different projects I've been participating in. One, to develop a pool of synthetic reference samples for microRNA profiling, and the other to look at microRNA expression levels in patients with broken bones. Next slide, please. Next slide. So first, just a few slides about what microRNA is, for those of you who might not be so familiar with it. Compared to messenger RNA, which is, of course, the intermediate between the code of DNA and the eventual end product of protein, microRNA are very short species of RNA, typically 18 to 22 nucleotides in length. And those nucleotides have sequence composition that partially matches target sequences within messenger RNA. So the eventual function of a microRNA is either to block protein translation by serving as a guide sequence to recruit proteins that block the translation, the ribosomes on the messenger RNA, or to actually degrade the template mRNA. MicroRNAs are present in most cell types and in circulation for most mammals. And normally, microRNAs are found throughout development and in normal physiological processes. And they're also often show altered levels of expression under pathological conditions. Next slide. Next slide, please. If you're interested in exploring more about microRNAs, miRBase is a good place to start. miRBase is a catalog of about 28,000 different microRNAs among a number of different species that have been cataloged in miRBase. And if you just look at the human microRNAs, there are over 2,500 of them. Now, that's certainly lower than the number of human genes, but because microRNAs are redundant in that they target several different genes at possibly at the same time, it's thought that a number, if not most, of the human genome genes are under potential regulation by microRNAs, even though there's only about 2,500 of them. while it's easy to discover microRNAs by sequencing or other means, target prediction is a little bit more difficult. Because of the redundancy and the partial sequence complementarity, there are a number of algorithms out there for predicting what genes might be regulated by a microRNA. And rather than trying to list comprehensively all of those tools, I've just shown you the first page of a Google search for microRNA target prediction. And if you're interested in that, you can explore these resources further to see how targets are either predicted de novo or in silico, or are cataloged in databases of experimentally confirmed interactions between microRNAs and their messenger RNA. Next slide. So there are a number of different platforms for profiling microRNA, and they range over the typical, molecular platforms for studying RNA. From RT-qPCR, where you're looking at just one microRNA at a time using traditional quantitative PCR amplification primers and perhaps a probe, hydrolysis probe detection like the TACMAN system, through multiplex systems, which could include something like a Fluidigm multiplex PCR platform or other multiplex assays like Luminex or related nanostring types of platforms. Here you would detect a set number of microRNAs all at the same time. These typically range in the hundreds of microRNAs, although, for example, you can buy TACMAN gene cards that cover with 2 cards the entire complement of human microRNAs. Then we'd move up again in scale to microarrays, which would be composed of all known microRNAs, either for one or a few species or for all species, as in the case of the Affymetrix microarray. Here, the assay is typically based on hybridization of the labeled target sequences to immobilized probes. RNA-seq is a very powerful platform for doing deep sequencing. Here, you can not only map the sequencing results to known microRNAs, but also discover microRNAs in the short size range that perhaps haven't been observed before. And then today we'll spend a lot of time talking about the HTG molecular EDGE-seq assay. The main differentiating conditions about EDGE-seq are in the sample source column. You can see that most assays typically require purified total RNA, and in some cases can also be used to work with purified microRNA that has gone through size selection. And the purified total RNA is typically amplified by either pre-amplification or quantitative PCR. And in the case of the TrueSeq protocol for Illumina deep sequencing, the cDNA is gel selected to select the size range for microRNA, and then that's used to feed into the Illumina library construction process. The input amounts for RNA profiling typically range from nanogram amounts for PCR-based method up to a microgram for deep sequencing of total RNA. And as I mentioned, the coverage can range from one target to everything known and novel in a sample. For EDGE-seq, as I said, one of the differentiating factors is the input. Here, EDGE-seq profiling can use a crude lysate from tissues or cells. In fact, that's the preferred source. So as you can see listed here, very small amounts of samples, thousands of cells, a 5-micron section from FFPE tissue, a few microliters of either serum or whole PAX gene preserved blood can be used to profile, in the case of the HTG platform, all known human microRNAs. The basis of the assay is a nuclease protection followed by deep sequencing detection. And I'll talk about a little bit of that on the next slide. So first, let's look at the workflow. The initial step is to lyse your tissue or cell sample. No RNA extraction or purification is required here. That then feeds into the EDGE-seq platform, in which an automated robotics unit will perform the nuclease protection assay, in which synthetic DNA probes to known microRNAs are used to hybridize to the sample. And then any mishybridized or non-hybridization bubble is digested, and that's the basis of the nuclease protection. Then any surviving probes are fed into library construction, and those surviving probes are sequenced on an Illumina platform either a MiSeq or a HiSeq sequencer. Next slide. The gritty details of this assay are shown here, where we are using synthetic DNA probes, the nuclease protection probe, which will hybridize to any RNA target, either micro or mRNA, depending on how you design the sequence. And then S1 nuclease is used to digest either unhybridized probes or any mismatched probes that are occupied partially by a RNA target. The surviving DNA probes then are used for sequencing. So here, the difference between this assay and normal deep sequencing is that what's actually being read here is the surviving DNA synthetic probe, not the endogenous RNA or cDNA resulting from endogenous RNA. A little bit more detail on the next slide, and these slides will be available archived on the GenomeWeb site on the next, sorry, if you go back one. Okay, I guess we lost one slide here, but the details of this assay are available on the HTG website. There are non-hybridizing wings on either side of the nuclease protection probe, and those are what are used to create the sequencing library. And the point here is that any digestion, even of a one base pair mismatch, should remove one or both of the wings from the rest of the probe template, and that means that that probe won't survive to be detected by sequencing. So this is a very sensitive assay that can detect down to a one base pair mismatch. All right, next slide. So the first project I'd like to talk to you about is in collaboration with the Association of Biomolecular Resource Facilities, ABRF. And what we've done in the Genomics Research Group is set out to try to develop standards for microRNA profiling. These reference standards are useful because they can allow you to fairly compare different platforms using the same reference sample. They can also be used to develop new protocols, because if you're going to improve either a wet bench process or a bioinformatics analysis, you'd like to be able to compare your new method to the same sorts of samples run previously. And then also of interest to ABRF members is the fact that standards allow you to compare how well your lab is doing over time with various client samples to improve your assay service. Next slide. So we designed these standards by first taking the entire miRNA-based database for human microRNAs and using clustal-W to align it. So what happens then is by assigning a serial number in the left column to these aligned sequences, we can pretty much summarize the full range of RNA composition in the microRNA pool for human. And by doing that, then we can assign these different microRNAs to different pools such that family members can be put into different pools. And we're also ensuring that we're covering the full range of sequence composition that's represented by microRNAs in human. So these pools were designed by randomly selecting across these serial numbers listed for the aligned microRNAs, creating one pool of 829, that's the background pool, and then 4 pools of 16 different microRNAs. And I'll show you why we selected out these subpools in a minute. This random selection process was done so that hopefully we're representing what a sample might look like if it contained the normal range of microRNAs expressed in any given sample. But of course, tissues and cells don't express all genes nor all microRNAs at the same time. So by having pools of 16, or in the background case 829, we're hoping that at least a few will be detectable by any given platform. Next slide. So I retained a few of the microRNAs based on membership in families, and those family members are indicated here with the differences in nucleotide sequence in red. So by putting different family members in different pools, what we're hoping to do is be able to see whether or not an assay can discriminate between family members. Because different pools will be present at different concentrations, they should give different signals, and that signal should be able to be picked up regardless of whether you're a family member in pool 1 or 2. Next slide. So the standards, 5 standards, A through E, are constructed by combining different amounts of the pools. The protocol for using these standards is going to be listed at the abrf.org website. And in fact, you can obtain these pools for free if you'll just send me shipping information. And my email address is located here at the bottom of the slide. So if you follow the protocol and create standards A, B, C, D, and E, what results is a Latin square design of the various pools at different concentrations. Next slide. Here are some EdgeSeq sequencing data for the synthetic pools that were performed at HTG. The black numerals indicate the expected signal for each of the standards, and then the lines and points are the experimental data. And as you can see, the EdgeSeq assay is working well, giving the expected amount of signal for each of the standards, depending on which pool composition was used to make those standards. Next slide. The EdgeSeq assay also was capable of picking out family members. So here, for example, on the left is 301B and 301A, which differ by just 2 nucleotides in the middle. And the expected signal is different for those 2 different probes in pools 3 and 4, and that's the case experimentally. The same for a 1-base difference in the case of the LET7A versus 7E probes. Next slide, please. Reproducibility was also good for this assay. Here, the pool 5, which is the background pool with
You're muted. I didn't hear you.
We lost the audio. We will fix this momentarily. Sorry about this.
Can you hear me? Hello?
Yes, we can hear you. Thank you. Please continue.
Okay, sorry about that, everyone. So I was just talking about the reproducibility of the assay. Here, the 5th pool that contains over 800 microRNAs was run 3 different times on 3 different days, and you can see the R-squared correlation values were very good for these replicates. Next slide. Then the pool was diluted to, as you can see, 500 femtomolar or 250 femtomolar, and again, reproducibility between triplicates was very good. So this is a sensitive assay that performs consistently over several runs. The next slide. So having established that the EdgeSeq assay for microRNA was working pretty well, another set of collaborators that I'm involved with at a startup company called MSKDX, wanted to test microRNA profiles in orthopedic trauma populations. So a little bit of background, about 10 to 20% of bone fractures don't heal. This is a condition called non-union, and it affects about 700,000 patients annually just in the US. Non-union can be treated, but the FDA guidelines are that you first have to wait for up to 9 months to see whether potential healing occurs before intervening. And the interventions can range from non-invasive bone stimulators up to revision surgery. But these, as I said, can't be attempted until after 9 months of watchful waiting. So we believe that an early test during the healing process might be able to give a prognosis of whether or not productive healing is occurring and allow patients that are going to be non-unions to get their revision surgery or other intervention much sooner and thus prevent months of waiting and comorbidity. Next slide. So here are some preliminary data from one of the healing time points, about 6 weeks after the fracture. And in the top and upper left, you can see either slow or non-healing patients' results on the EdgeSeq assay using whole blood preserved in PAX gene. On the bottom and lower right, then, are also the patients that were slow or non-healers, but using serum. And as you can see, there's not much overlap. And that overlap is not very much with purified RNA that was run on Affymetrix chips in the upper right. So the take home here is not only are we detecting microRNAs, but those microRNAs are, the profiles are quite different depending on the type of sample that you use, whether it's whole blood, purified RNA, or serum. Next slide. So we have a number of serum biomarker candidates that came out of this initial assay, and some of those representative results are indicated here, where green dots are normal healers, blue or purple is the non-union patients, and red is slow healers. And you can see we have microRNA biomarker candidates that are either expressed at a lower level in the pathological healing condition compared to normal healers, or are induced, expressed at higher levels compared to the normal healers. So we'll be following up on these biomarker candidates as potential predictors of whether or not productive healing is occurring. Next slide. So to summarize the first part of our webinar then, I just want to reiterate that deep sequencing detection of nuclease-protected synthetic probes, which is the basis of the HTG EDGE-seq assay, clearly supports microRNA profiling, and it works pretty well from crude samples, lysates of small amounts of tissues or cells. Also, I've introduced a Latin square set of synthetic reference pools that can be used to measure the dynamic range and response of any given platform, and also can be used to detect specificity of detection of different microRNA family members. And then finally, we've shown that serum microRNA profiles give a slightly different or very different result from crude cell lysates or from purified RNA, at least in the case of our orthopedic trauma population, and that this serum might be a convenient and clinically relevant source of biomarkers for the fracture healing process. All right, that's the end of part one, and thanks for your attention.
Yeah, thank you very much, Dr. Baldwin. So just as a reminder to our webinar participants, if you have a question, please type it into the Q&A box in the control panel, and we will conduct the Q&A portion of the webinar after the last presentation. So our next speaker is Dr. Scott Tebbett from the University of British Columbia. Please go ahead.
Hello everyone, Scott Tebbett here. So I will be talking for the next few minutes about the Proof Center of Excellence and its biomarker programs and some pilot work associated with acute heart failure recovery. Next slide, please. So the outline will just be talking a bit more about Proof Center, acute heart failure and its treatments, and then our pilot microRNA biomarker program in acute heart failure. Next slide. So the Proof Center of Excellence is a not-for-profit organization located at St. Paul's Hospital in downtown Vancouver, British Columbia, Canada, and we're hosted by UBC and Providence Healthcare. And our mission is to develop high-value biomarker-based blood tests by harnessing the integrated power of clinical, molecular, and computational science. Next slide, please. So there are a lot of processes and competencies involved in what we do, and we collaborate a lot with other organizations internally at the hospital and externally with organizations such as HTG Molecular. And over the past 10 to 12 years, our Proof Center has established a clinician-driven end-to-end biomarker development process. And importantly, shown on this figure, we start with the clinical need and an economic analysis. You know, what if we were trying to develop biomarkers that we can implement into the healthcare system, what really is the clinical need before we even do any biomarker discovery and content development? And eventually, we need to go through the whole validation and qualification process, make sure the assay is effective, day-to-day, week-to-week, month-to-month, year-to-year, across different sites around the world. So an awful lot of different processes, competencies must be followed in this process. Next slide, please. And our portfolio of biomarker programs involves anything from in the transplantation world for acute heart and kidney transplant rejection, and all the way through to lungs and different programs in heart, and I'll be talking about acute heart failure in a little bit more detail, and also in kidney. Next slide, please. So for acute heart failure, acute heart failure syndromes represent nearly 60% of direct heart failure costs in North America, with mortality more than 50% at 60 days post-discharge. Now, mechanical circulatory support, either long-term or implantable for long-term, is sometimes an essential intervention to keep these patients alive in the absence of transplantation. And our task is really to discover blood-based biomarkers to guide the management of patients with acute heart failure. So effectively what we're looking for is to be able to predict survival from acute heart failure, also to identify patient responses to these mechanical circulatory assist devices as therapies, and also to stratify responders versus non-responders to such device therapy. Next slide, please. So let me talk a little bit more about these mechanical circulatory assists. So one such assist is a ventricular assist device, or VAD. So implantation of VADs is a treatment modality in heart failure cases with failed inotropic response in the presence of organ hypoperfusion. And it's been shown that it can improve survival rates. So these VADs can be used as a bridge to recovery, a bridge to transplantation, or even as destination therapy. And the implantation of these VADs has been shown that it can allow recovery of cardiac function through chronic unloading of the heart, has been shown to normalize biochemical and functional parameters in the cases. They're not that common, but there are cases of recovery, and this is through reverse remodeling. So effectively, you implant the assist devices into someone with heart failure, and it basically takes the strain off their heart by allowing a mechanical circulatory system. Next slide, please. And this is shown a little bit more in this slide here. So this is an example of one such mechanical circulatory assist, the HeartMate, and it basically, effectively, the surgeons are plumbing this device into the patient's heart. It goes into the apex, or the very bottom tip of the left ventricle, and then it comes through an impeller. So the blood, instead of being pumped out of the left ventricle into the ascending aorta of the heart, it actually it's pulled out of the left ventricle through this impeller device, and then it gets put back into the circulation at the aorta. And what this can do is unload the heart from its pumping action. And these people, because this is a continuous propulsion mechanism, the people who have these things in them effectively don't have a pulse. So they go around walking around the world without actually having a pulse. But they do have to carry batteries and spare batteries, obviously, with them. Next slide, please. So our acute heart failure program, the experimental design is basically people come into the hospital with acute heart failure. They obviously have to be consented for research studies, and then we're allowed to take either blood samples and or a myocardial biopsy sample at the time that they go into the have an implantable ventricular assist or mechanical circulatory assist. And then subsequently, several days after implantation of this device, we can obtain peripheral blood samples, and then on a weekly basis, and then eventually these patients will either go on to 3 different things. They will die, they will recover, or they will go on to transplantation. And at the post-device sample collection, we can actually collect, again, blood samples, and in the case of where the heart is removed, we can get another tissue, a myocardial tissue sample. Next slide, please. I won't dwell on this because Don's very nicely gone through the HDG-HSeq system, but just to look on the upper left-hand side of this slide, just shows the PAX blood tubes that we use and a slide with a formalin-fixed paraffin-embedded tissue. So those are the sorts of biospecimens that we're actually using here. So peripheral blood through PAX tubes and formalin-fixed paraffin-embedded myocardial cardiac tissues specimens. Next slide, please. And keep going. One more slide, please. Thanks. We've already gone through this. Thanks. So let me show you some of the data that we've obtained in our pilot work. So in the top panel A, the 3 panels there are effectively showing the coefficients of variations across different sample types that we have. And then on the x-axis is the normalized count of the microRNAs that we've been detecting. So you can see on the y-axis the threshold is 10% CV, and most of our microRNAs that we're detecting in the different tissues falls well below the 10% threshold for that. So that's good to know based on technical replication. And the first 2 panels are based on the formalin-fixed paraffin-embedded samples from Apex. Apex is basically the very bottom tip of the left ventricle, which is where you plumb the device into, so that allows the surgeon to take a bit of tissue sample out of the heart. And then the middle panel there is the left ventricular free wall, which I'll be talking about a little bit more in the talk. And the left ventricular free wall is again on the left ventricle, but it's further around on the left-hand side. So the free wall that's expanding quite a lot during heartbeat if it was beating effectively. And so we can obtain that sample type rather than going back to the apex, because there's a lot of injury at the apex when we remove the heart. And then the final panel on the right-hand side is the showing the coefficient of variation across detectable microRNAs in peripheral blood. And in panel B, the lower part, it just shows you the overlap between blood and cardiac tissue in this Venn diagram, showing that we can detect a reasonable number of microRNAs, and then there's a substantial overlap of these microRNAs between peripheral blood and the tissue section. So whether these things are related to immune cells that have been infiltrating the heart or other type common pathways that are controlled by microRNAs, that's for our further work. Next slide, please. So this is our pilot study, our design here. So I'll walk you through this, and I'll show you some data from each of these 4 contrasts of interest itemized on the left-hand side, and also shown in the red arrows on the right-hand side. So the first comparison we did was look at the left ventricular free wall between normal medically managed patients with heart failure versus the left ventricular free wall tissue specimen in patients who had an implantable ventricular assist device. And this was obviously at the time of explantation of heart. So these subjects would have gone on to transplantation. So we looked at that effectively to ask the question, are there differences in microRNAs between medically managed heart failure and VAD managed heart failure? Our next contrast of interest was number 2, where we just looked at normal hearts. So these are effectively from deceased patients who were allowed to obtain some heart tissue. And we're really comparing left ventricular free wall versus the apex tissue specimens in the heart, really to show that there was no site-specific differences in these collection sites. The third comparison, number 3 here, was in the heart failure cases again, just comparing the patients and in the apex versus left ventricular free wall. So again, the apex specimen is taken at the time of implantation of the ventricular assist device, and the left ventricular free wall, or LVFW, sample is taken at the time of explant, or at the time of the transplantation itself. So this is effectively, you could construe it as a treatment effect of the ventricular assist device itself. And then final comparison, number 4, is effectively peripheral blood samples in before ventricular assist device implantation and post. Just really to see if we could detect anything that we saw in the heart tissue, could we detect this in the blood? Next slide, please. So the next 4 slides are going to be basically little volcano plots showing the log fold change on the x-axis with the statistical p-value on the y-axis, and anything that survives a false discovery rate of about 10% or even 5%, I think, is in blue here on the left-hand panels of these volcano spots, and it's just a clustering diagram of these particular discoveries on the right-hand side. So you can see this is just really comparing the first contrast, which was medically managed versus ventricular assist device managed heart failure patients, and showing that we can detect differential expression in microRNAs in heart tissue between these two patient groups. Next slide, please. And then this is our sanity check here, because basically we were trying to look between in bad treated patients. We're looking for a comparison between two different sites of the heart itself, purely because of practicality reasons, not being able to go back into the same site once this VAD has been plumbed into the apex. So here you can see a similar comparison, but there's very much lower discovery rates here for microRNAs, showing that there probably aren't that many differences in microRNA expression, at least between left ventricular free wall and apex. And also note that on the x-axis, the fold changes of these microRNAs is very much reduced compared to the previous slide. Next slide, please. Now this was our comparison, basically pre-VAD versus post-VAD, and you can see here, a lot of discovery coming up here in the blue highlighted microRNAs. And of course, this is a paired. We can do a paired test here because they're from the same patients. But you can see there are an awful lot of microRNAs that are differentially abundant between patients before they've received their ventricular assist device and post. Of course, the effect of surgery itself needs to be taken into account, and we have to follow up with a lot of these discoveries. Next slide, please. And then this is our final comparison of interest, where we could actually see, is there anything in the blood that we could detect? Obviously, we don't want to be taking biopsy samples from patients just to have a biomarker test. Patients generally, heart failure patients or heart transplant patients, absolutely detest having a biopsy where a catheter is put into their heart and a chunk of their heart is removed for pathology purposes. And they, as you might imagine, they don't like this type of thing. It's very, very invasive. So what we hope is that we can find microRNAs and then look for these in the blood, and then develop the blood signatures as biomarker tests themselves as a simple blood test. And you can see that obviously not so much discovery here, but there are a few microRNAs that are differentially expressed in blood samples between pre and post ventricular assist device. Next slide, please. And some of the interesting microRNAs that we found, certainly in the cardiac tissue, so microRNA 145-5P has actually previously been reported upregulated in patients in the plasma with a positive left ventricular reverse remodeling following cardiac resynchronization therapy. And also microRNA 143 and 145 have been implicated in other studies as to modulating the vascular smooth muscle cell phenotype in response to injury, including the aorta and media and intima tissues. And microRNA 199A is particularly interesting in our pilot data. Next slide, please. So these three plots here show the comparisons between just one of these microRNAs that we've been looking at. And on the y-axis is the abundance of, or in counts, log2 score of this particular microRNA. So you can see that this shows that we do find this 199A5P differentially expressed between heart failure and ventricular assist device, and also it's differentially expressed in the longitudinal samples of the blood itself. And it's been known for a while that this microRNA is mainly expressed in lung and heart tissue. It's inhibited in response to hypoxemia, and it's been shown to be upregulated in hypertrophic rat hearts, and even been recently reported to induce cardiomyocyte proliferation in a rat model. And so, these are only pilot studies. You can see our sample sizes are very low here. But if you go on to the next slide, we're hoping to do a longer study here where we can actually really get to find potential biomarker panels here that could be useful. And so, we're obviously recruiting these sorts of subjects in the heart clinics can be problematic, but we've now built up a sample size. I will be comparing medical management, about 45 patients, with implantable VAD patients, again 48 patients, sorry. And so we can study a lot of the samples that we're obtaining from our surgical colleagues here at St. Paul's Hospital. And next slide, please. So in final, I'd just like to acknowledge a lot of the research participants who donate their specimens here, the nurses and coordinators who track these patients down and draw their blood specimens, Anson Shung and Kristen Ramanthan at St. Paul's Hospital, who are the surgical cardiologists who do obtain a lot of the specimens, Bruce McManus at UBC and Proof Center, and also the people that did a lot of the analysis, Anna Meredith, Casey Shannon, Darlene Dye, and Yuri Yang at the Proof Center, and also thank Deborah Thompson, Ehab Putros at the HTG Molecular, and also some colleagues at the Scripps Research Institute down in San Diego who kindly sequenced the libraries for us, Steve Head and Tony Mondalo. And next slide, and thank you very much for your attention.
Thank you very much, Dr. Tebbett. So again, as a reminder to our participants, please type your questions into the Q&A box in the control panel, and we will ask your questions after the last presentation. So our next and last speaker is John Lucky from HTG Molecular Diagnostics. So please go ahead with your presentation. It looks like we lost the audio again. Please give us a moment. Thank you. John, can you say something?
Can you hear me now?
John, we can hear you now. Please go ahead.
Can you hear me now? Great. Apologize. Apparently I have a loose wire on my phone. Thank you first. I'd like to just start off. I'll keep my part short and sweet because I know there's been some excellent questions coming across for Scott and Don. First, I'd just like to thank them both for sharing their exciting research with us. I think they're both really excellent examples of what the HTG EdgeSeq microRNA platform can do for you. We are a small company in Tucson, Arizona. We always like to show this picture, especially this time of year, and it's one of the benefits of living in Tucson, where it's nice pretty much all the year, especially in December. Next slide, please. Before we continue talking about the microRNA, I just want to point out that we do have mRNA panels for the EdgeSeq as well as the microRNA. Just real briefly, we have the EdgeSeq Immuno-Oncology panel, which is 549 immuno-oncology-related genes. Like the microRNA panel, this is optimized to work with extremely small amounts of sample input, 1.5 millimeters of FFPE tissue or a few hundred cells. Its larger cousin is the HTG EdgeSeq Oncology Biomarker Panel. In this case, it's 2,560 genes, which are all, again, typically oncology-related, but also cover many other systems within the body and the cell, allowing for a broader view of what is happening with it, whether it be a tumor or a blood specimen. And the last assay I'll mention just briefly is our HTG EdgeSeq Lymphoma panel that we just launched. This panel is really useful for lymphoma typing and analysis. It may be useful for some of the folks on this call, as I've seen look through the institution that you're at. Next slide, please. So as we've alluded to in both the other presentations, all the data that we presented today was run on the HTG EdgeSeq system. This is an instrument platform. It's a benchtop instrument, approximately 2.5 feet wide. It automates the Q&PA process that both Scott and Don have alluded to. It's a very simple, easy-to-use instrumentation where the samples and the pre-configured reagents are loaded into a series of drawers. There is a PC-based user interface which the sample information is loaded up into. Essentially, once the samples are staged and the instrument cross-checks the reagents that are placed within it, you press a button and you come back the next morning to your processed samples that are then ready for the library preparation. Next slide, please. For the microRNA kits that both Scott and Don have used, we actually have them available for both Illumina and Ion Torrent sequencers in multiple different configurations. We offer these different configurations for two purposes. Obviously, we want to make sure that we have the proper throughput for you. So if you have a small number of samples to run at a time, we have kits for that, as well as the larger 96-well kits. In addition, we also have kits that will maximize the amount of sequencing that you can do on the various different sequencers. Really, the best way to identify what's the best kit for your sequencer solution and your needs is to contact our sales representatives. And the next slide. Hopefully, the research that you've seen has convinced you that you want to try out the HCG microRNA whole transcriptome assay. We have two ways of doing this. The first is we do small proof-of-concept studies in our in-house service facility. The second way we can do this is with our instrument performance evaluations in your facility, so where we would stage an instrument and work with you to set up a pilot study in your facility. This is typically better if you have a larger amount of samples. Regardless of how you want to go about trying the assay, the first step in contacting us is to contact our local HCG business development representative, and we can schedule the evaluation and really figure out what the best way to get your questions answered. The easiest way to reach us is at the info@hcgmolecular.com email listed below. And next slide, please. I believe that's the final slide. So at this point, I'll hand it back to Julia. Thank you again for moderating the webinar, and we appreciate your time.
Yeah, thank you very much, John. So before we enter the Q&A, we'd just like to ask our attendees to please take a moment after the webinar has ended to take our exit survey to give us your feedback. We will now start with the Q&A. So let's start with some questions for Scott. So one participant wants to know why you need to use mRNA as biomarkers in your cardiac study. Why couldn't you use other gene? Wouldn't it be easier to quantify other genes rather than miRNA? And is there any evidence showing that miRNA are better biomarkers than other types of genes?
Yeah, thanks. Very great question, please. Thank you. I think, I should say from the outset that we're trying to cover all bases here. So we've been doing a proteomics biomarker study where we're looking at protein expression in the tissues as well as in blood plasma. We have a publication led by Anna Meredith, whose PhD was based on this program of work. So we have a publication in press on that, so with the proteomics. We also are interested in messenger RNAs. There's a lot more annotations, obviously, associated with messenger RNAs than microRNAs. So, we are trying to do a sort of, we are not wedded to any particular molecular type. We really, we try and create our biomarkers based on what the data tells us. Having said that, microRNAs have been shown to be fairly stable, so especially in peripheral blood. So, we prefer to use something that, where the specimen type, SOPs relating to biospecimen handling are fraught with issues. And, when you're really trying to put a test into a healthcare system, you have to be able to have an assay that's robust to, slight differences in specimen procedures. So we think that microRNAs probably might be a little bit more robust to that than the messenger RNA, certainly for storage of samples. So I can't answer your question precisely at the moment, but that we are trying a variety of different techniques.
Yeah, great. That answers some of it. Another sample-related question, did you find any issues with the protocol or with the results when you used FFPE samples compared to blood samples?
No, not really. We had a few of our samples failed our QC at the data analysis step, but these were spread out between the formalin-fixed specimens and the PAX blood specimens, so it didn't seem to be associated with that. There were no specific differences. We used lysates from each of these specimens, so we didn't purify the RNA.
Okay, great. Yeah. And here's more of a clinical question. Do you have any indication as to how long you should follow medically managed patient blood mRNA levels to determine if they've stabilized or if a VAD is required?
Now, that's an excellent question, and we'd have to go back to our clinical collaborators, and we'd have to go up to the heart clinics and consent some people with heart failure and actually take specimens appropriately to answer that question. And we could probably borrow samples from other studies, but we'd have to set up the entire design from scratch to answer that question. Yeah.
Okay, great, thanks. We have a couple of questions for Dawn. So one relates to the standards. So would the miRNA standard be used as a spike-in standard TruSeq mRNA-seq protocols and be distinguished from the endogenous miRNA? Sorry about that.
These synthetic microRNAs were taken directly from miR-based sequences. So to use it as a spike-in, you would want to be using it in a sample that doesn't contain human microRNA. So if you're doing a plant or some distantly related organism to human, it might be a spike-in. But if you're in a mammal, you're going to see a lot of overlap between the synthetic sequences and the endogenous ones. Now, having said that, the pools 1 to 4, which are the subset pools that only contain 16 microRNAs, might be useful as a spike-in, in that if you've determined that those 16 microRNAs are not critical to your results, you might be able to use just that one pool as a spike-in. Spike-ins are quite useful, of course, to follow the quality of your samples, especially when you have a large cohort of samples across the entire study. But I think if you were going to do that from scratch, you might want to design spike-ins from some sort of alien sequence, something that would look like a microRNA but wouldn't be represented in the genome of the organism you're interested in. Okay,
thank you. Another question for Dawn. One of the attendees is wondering about the variation in miRNA expression that you saw in your non-union bone study. It almost appears as though the expression depends on what function you're assessing, trying to assess, like the cell lysate for the cellular component or the serum for free markers. So do you have any thoughts on that?
That's correct. So we were also interested in that very question, and that's why we ran the same patients on both serum, whole blood, and on purified RNA. And it's clear that you can get different profiles depending on the sample type. Now, I should point out that those results were differentially expressed microRNAs. If we did the experiment like Scott showed, where it was just what microRNAs are detected, we would also see a lot of overlap between the different sample types. But it's the differential expression that really showed differences between the different either acellular, crude lysate, or purified RNA profiles. So I think this is a good cautionary tale for anyone considering these kinds of experiments, in that the way that you treat your RNA, the source that you get it, even if it ought to overlap quite a bit, can have a major influence on the profiles that you observe.
Okay, thank you. So here's a question for John. So is the HTG-EDGE-seq system applicable from sample to library, or is it from sample preparation to extract DNA?
Yeah, so it is a no extraction system. So when we process the sample, the kits include the initial reagents that we use for lysis. That crude lysate is then what is placed on the HTG-EDGE-seq processor system that you've seen the images of. That instrument will then take that sample up into the point where it's ready to be barcoded for subsequent sequencing. We provide those reagents, but we do not provide the sequencing reagents themselves. Hopefully the no extraction and the ease and the quality that you get with a no extraction assay shows through.
Great, thank you. And another question for you, John. So are there other HTG panel kits that use the same chemistry or the same strategy as the miRNA kit, like the nucleus protected probe?
Yes, that's correct. The other three panels that I showed you are targeting mRNA, but they use the same general concept. The only difference is slight differences in the processing that the edge processor does, and mostly around the temperatures involved. But yes, they are nuclease protection probe-based assays.
Okay, here's a very specific question for Scott. So what was the FFPE yield for miRNA?
I'm not sure how to answer that question. What the questioner means by yield. We didn't extract we did not extract RNA from the specimens, so we can't measure how much we obtained as far as yield of RNA or even microRNA. One of the slides showed the Venn diagram, so we can detect in these samples about 150 to 200 microRNAs at above background level. So, that's another way of answering this question. Hopefully that addresses the questioner's remarks.
Okay, thank you very much. One more question for Scott. So you talked about wanting to develop a blood signature as a biomarker test, and was wondering what's your next step to develop such a non-invasive test?
Good question. Thank you. I think what we need to do is, we have our biobank is growing, all these specimens in there. We need to now, go into that biobank and design a biomarker discovery study with a good sample size so that we can actually detect these things. Once we have a sets of microRNAs that are differentially abundant between our different contrasts or groups of interest, what we then do in the proof biomarker pipeline is to put that through a series of univariate and multivariate tests and classification algorithms such as elastic nets, random forests, those sorts of things, or linear discriminant analysis. That provides us with panels of markers, because usually you don't just get one marker or one gene or molecule that can distinguish a subject and put them into a specific category. So you usually have panels with multiple analytes. We then, we generate these things. You do all the receiver operator characteristic type analyses and positive predictive value, negative predictive value, all those types of things, using very deep cross-validation of your data set. Once you show that, yeah, we do have a classification signature for a fit-for-purpose biomarker tool, we then have to replicate that in additional independent subject samples. So, once you have that, then you can say, okay, now we need to sort of start getting it more further down the road of analytical validation and then clinical validation, and ultimately, probably a few years later, into clinical utility tests. So, this is only just the very beginning of a biomarker program.
Great, thank you so much. So a similar question for Dawn. What are your plans for developing the biomarkers for your productive healing further?
In a nutshell, ditto. We are also working on a large cohort of fracture patients, looking at not just at one time point, but at the entire healing time course, from a few weeks after fracture to a few months. So what we need to do is, again, perform these computer learning algorithms to find panels of biomarker candidates that may be indicative of slow healing or non-union, and then validate those on an independent cohort. And we have also been working with messenger RNA profiling, serum protein profiling, and microRNA profiling, hoping to come up with a multiplex set of biomarkers that will be robust across a large population of people. So we will also be, validating those biomarkers in small experimental sets and then translating that to a larger trial of the biomarkers in independent samples, hopefully leading then to a clinically useful test.
Thank you. So we have no further questions. So we would like to thank all our panelists today, Dawn Baldwin, Scott Tabbitt, and John Lucky, and also our sponsor, HTG Molecular Diagnostics. As a reminder, please look out for the pop-up survey after you log out to provide your feedback to us. And if you missed any part of this webinar or you wish to listen to it again, a link to an archived version will be emailed to all attendees. Thank you very much for joining us for this Genome webinar.